So, hello everyone. Hope everyone is doing good. Welcome to the PAPERI's basic level polygenic scores course. My name is Mariam Youssef, the PAPERI Education TA and today's moderator. Thank you for joining us for our sixth session on the introduction of polygenic scores. I'm delighted to introduce our esteemed speaker, Dr. Burja Darst. Dr. Darst is an assistant professor at the Fred Hutchinson Cancer Center. Her research is focused on identifying and understanding genetic and multi-omic risk factors of prostate cancer in diverse populations. She has extensive expertise in genetic epidemiology as well as genetic association and metabolomic investigations. Her research focuses on understanding genetic risk of prostate cancer in multi-ancestry populations, particularly using rare variants captured with whole exome sequencing, genome-wide association studies, polygenic risk scores, and metabolomic to distinguish aggressive from non-aggressive disease and to understand the stark health disparities that contribute to prostate cancer risk. She has also done research on genetic testing and precision medicine, developmental neurogenetics, and the genetics of complex aging traits, and worked to integrate genomic and metabolomic data to understand the underlying mechanisms contributing to complex aging traits and Alzheimer's disease. Currently, she's leading an effort to improve understanding of the genomic and metabolic mechanisms contributing to prostate cancer risk in men from diverse populations, as well as improving the utility of polygenic risk scores and rare genetic variants for the prediction of prostate cancer across patients' population. Dr. Darst has received multiple awards for her work, including the Prostate Cancer Foundation Young Investigator Award and the AACR Next Gen Star Award. Please join me in welcoming Dr. Portia Darts. Please note that this session will be recorded. Please mute your mics. Dr. Portia, the floor is yours.
Thank you for that introduction. And thank you all for having me today and this opportunity to give a lecture and talk about this topic. I hope people already had a chance to look over the lecture, and today we're having the Q&A session. So I think the first thing we can do is just open up the floor for any questions. Did anyone have any questions after watching the lecture or anything that they would like to talk about, even if it isn't really a question? Yes, Oscar?
Thank you very much. It was a very interesting lecture.
Excuse me, Oscar, please introduce yourself and your country, then ask your question. Thank you.
Thank you very much. My name is Oscar Nyangiri. I am a genetic epidemiologist. I'm based in Makerere University and in Uganda, and also a visiting lecturer in Kisii University. So thank you for the lectures. I just have a question about using only a few variants for polygenic scores. I don't know whether it's possible or not to use only a few variants. I noticed for the pancreatic cancer, you had the 8q, there was a variant that had quite a big effector.
Yeah.
Prostate cancer, sorry. So I'm wondering, are we able to just take only a few variants because of resource limitations and then try to create a case control study just to identify a few variants? I don't know what your take on that is.
Yeah, it really depends on the outcome that you're studying, like how strongly genetic is it, and are there known risk factors for it? So the outcome that you're studying, like how heritable it is, and what's known about that trait in terms of the genetic risk factors. And I think also I didn't talk about the different. There's like genome-wide approaches to constructing polygenic risk scores, which I didn't really go into too much in this lecture, but for some traits, there is evidence that if you include more variants across the genome, it becomes a better predictor of the trait that you're trying to study. So often they're called genome-wide polygenic risk scores, and they will include like 1.2 million or so genetic variants. And for some traits, those are really good predictors, better than just including, the few variants that are actually significantly associated with the outcome of interest. That's been shown to be a better predictor for traits like cardiovascular disease and diabetes, and I think it's been looked at for breast cancer too. But we looked at this with prostate cancer recently in the past couple years, and we found that it wasn't the case that including more variants for prostate cancer was a better predictor. We saw that those genome-wide approaches do about as well as just using the variants that we know are significantly associated with prostate cancer. So for prostate, we have, at the time that we did that study, it was 269 variants. So it's not too many. It's maybe still more than a handful. But in that case, our 269 variants worked as well as a genome-wide approach that used 1.1 million. So it's a much, much more like not as resource intensive to only have 269 variants that you're genotyping, but I can understand that even 269 could be a lot in some instances. I think in that case, that is something that we're thinking about, whether, at least in the case of prostate cancer, if there are a subset of those 269 variants that are really the most predictive of prostate cancer and the others are, important but not really contributing to the core association that we see. So I think this could be true for a lot of diseases, and maybe more research would need to be done to see kind of like what is the minimum number of SNPs or genetic variants that could be genotyped to still have a polygenic risk score that is pretty predictive. But what you mentioned with AQ24 is interesting, and I think it's kind of unique to prostate cancer that there are some variants that on their own, just one or two variants have really strong associations with prostate cancer. And I think those, you could look, there are a handful of variants that you could look at on their own without a polygenic risk score and still have a pretty good predictor of prostate cancer and disease aggressiveness. And I think that AQ24 variant is a good example of that. I can maybe just show that I have it here so I can show if people, for those who might not have remembered that part. So maybe I'll start with this slide here. So this GWAS was conducted a few years ago, where we identified, sorry about that. I don't know if you could hear that. Let me share again, and I won't go into presenter mode. Okay. I hope you can see this okay. So here, this GWAS was conducted a few years ago where they identified this really strong association with prostate cancer risk, and the odds ratio for this one variant is 2.3. And this is really an odds ratio that we're often looking for with a polygenic risk score. So this one variant is doing a lot and really could be useful on its own for prediction of prostate cancer risk. This is just looking at the development of prostate cancer, so incident prostate cancer, but we also found that it was found at a higher frequency in those who had more aggressive disease. So here on this figure, the further right you go, the more aggressive the disease, and you can see the odds ratio goes up a little bit, and the frequency of the allele, the risk allele, goes up in those with more aggressive disease. And then when we found that when we look at a polygenic risk score, at the time, there were 180 variants that were known to be associated with prostate cancer. When we remove this one variant in the AQ24 region, you can see there's a pretty huge drop in the predictive ability of the polygenic risk score, just suggesting that this one variant alone really is driving a lot of the predictive ability for the polygenic risk score. So I think there could be some instances of a handful of variants being really predictive, but it really depends on the disease and what we know about that disease. If we hadn't identified this AQ24 variant, then we wouldn't be able to do this. But I think there are some examples where a handful of variants could be really predictive of disease risk.
Thank you very much. That's very helpful.
Yeah. Thank you for the question. Any other questions from anyone? Any things that you were thinking about or wondering about as you were going through the lecture? Anything that was unclear? Yeah. Rachel?
Yes, please. Good morning. My name is Rachel and Rachel Osage. I'm a PhD candidate in McGill University, and I'm currently working on a project that has to do with developing and validating polygenic risk score for a pain phenotype. Okay, so my question is, I see that from my little experience, I see that, most of the number of SNPs that are required for me to um, construct my polygenic risk score is in thousands. So I'm wondering, is there a way to know the optimum number? Because I'm at the point where I am trying to improve my score, my performance, so to say. So at the moment, I'm having a performance of about 0.58. So I'm trying, to probably use meta-analysis, combine, more larger data sets, and I'm asking myself, would there be any um, change by increasing the sample size, the number of cohorts I'm including in the discovery data? Is there going to be any difference? The reason I'm asking this is because, is there a way to know, how many SNPs really would be useful for a particular phenotype, instead of just more like doing series of tests and eventually you find that, okay, this is what it takes for this phenotype. You can't go beyond this. So that's my question. I don't know if it's very clear. And are there
Yeah.
computational tricks, to improve, a PRS? Initially, I had used the clumping and thresholding approach. I tried to use the Bayesian PRSCS,
Mm-hmm.
and still there's no much difference in the performance. So I'm wondering, is it that it's the peculiarity of the phenotype, or there's a way I can, theoretically or, conceptually, estimate how many SNPs will be required in my final PRS, so that I estimate how many more cohorts I need to look for to get
Yeah.
that, to achieve that goal.
So what is the outcome that you're looking at?
It's a chronic pain phenotype. I'm
Oh,
trying to,
okay.
yes,
Okay.
yeah.
Okay. Yeah, when I was in graduate school, I did a lot of work looking at cognitive function. And I don't know how much is shared between chronic pain and cognitive function, but I felt like it was a very hard trait to study the genetics of because it's hard to measure. So I imagine that you could have a similar problem like that too. But I definitely think that the bigger the sample size, the better. As big as you can have for your discovery will definitely
Okay.
help with your polygenic risk score. And I think the question about how many variants, it's a really hard one to answer, and I think it's different for different traits. But one thing that you could do is like when you're using PRS CSX, like you mentioned, so there's PRS CS, which is designed for looking at one population at a time, and then PRS CSX is a newer version of it that will look at multiple populations at a time. And the recommendation for PRSCS is or PRSCSX is to use there's like 1.1 or 1.2 million HapMap3 variants. I don't know if you came across that when you were using it.
I,
Did you?
I I
Okay.
I came across that, but I wasn't sure because I'm still at the stage where I'm getting used to the software. The
Yeah.
authors say you must use RSIDs, and what I have in the base position chromosome and the allele 1 and allele 2. So I need to get the format into the form of that will be acceptable by the software. So I'm
Okay.
still at the experimental stage using, PRSCS to get the best of it.
Okay. Let me see if I can pull up some. I don't think I included any slides about this, but maybe I can find some slides to show to talk a little bit more about that question. But definitely the bigger the sample size, the better. Like that's one of the most important things you can do for developing a polygenic risk score is having a big sample size. And let me think of some where we would have shown things like this. So maybe this one. Okay, not that one, sorry. Okay. Oh, okay. I have some slides here. Let me just see if these are updated. Oh, no, I have recent ones. Let me see. Okay. Oh, okay. Here is one slide on this topic. Doesn't really go into it too much, but I'll show you this. Okay, so in this, this is a paper that we published last year where we were considering the question of like, of what approach should we use for a polygenic risk score. We're really wondering like, should we be using these million variants that are recommended by a lot of the genome-wide approaches, like you mentioned, PRS-CSX, which is this one right here. We looked at a number of other genome-wide approaches. Some of them you may have come across, like LD-PRED is one, EBPRS, these three others here. So we compared all of these different approaches, and all of these PRS in green are constructed using these 1.1 million HapMap3 variants. So you could, I would definitely recommend looking into the 1.1 million HapMap3 variants, because that, it kind of, then you don't have to kind of like guess at which variants you should be including. Like the pruning and thresholding is a good comparison. Like you could do that in addition to using the HapMap3 variants. And then in black, this is just showing all the variants that were significantly associated with prostate cancer at the genome-wide significance threshold. So you could try that too, like for chronic pain, just looking at the variants that are significantly associated and independent of each other. The one thing that we did here that I think was really helpful to improve how strong of a predictor this polygenic risk score was that we did statistical fine mapping. So once we ran the GWAS on prostate cancer, and we saw that there are these 269 regions that are significantly associated, whenever you see those peaks in a GWAS, let me see if I have a GWAS plot. This is one region, but there's so many that are significantly associated with prostate cancer. And it's hard to know, like you often will just go with the most significant one. But because of the correlation structure at each of these regions, that might not actually be the best signal to just use the one that's the most significant. So we do statistical fine mapping, where the goal is to try to identify which variant in this region is really the causal variant that's causally associated with prostate cancer. And that has a lot of limitations because it's statistics. So, there's always limitations, but using that approach to select the variants, the 269 variants among there were many more than 269 that were significant, but that's because of the correlation structure. But using that approach is one of the reasons why we think our 269 does really well compared to all these other genome-wide approaches. As in most other traits that look at these genome-wide approaches that include millions of variants, they often see that it does do better than a genome-wide significant polygenic risk score, but they don't do statistical fine mapping. So I think that's one thing that you could think about doing to try to improve your polygenic risk score without necessarily needing to have more variance. But you could do statistical fine mapping to try to better map the potentially causal variants, and see if that will lead to a better predictor.
Okay.
Yeah.
As a follow-up to what you just said,
Mhm.
what are your thoughts? What are your thoughts concerning the use of causal variants to improve polygenic risk score by way of a PRS set? That's the PRS SET, the software that tries to incorporate pathways and all of that. Do you think that that's another approach to improve the
Mhm.
PRS?
Yeah, so I haven't used PRS set, but it sounds like it's an approach that just takes into consideration like the function of the variance. Is that right? Kind of like functional annotation information. And I haven't used it, but I think it's a great idea because it kind of leverages a lot more than just these association results that you get when you run a GWAS, because it can pull in other information about like, there's all of this signal here, but are some of these variants more likely to be functional than the other just based on what type of a SNP they are? And that's a really important question too. So I think I haven't looked at those approaches, but I think it's a great idea and could definitely be an important way to try to improve the predictive ability of a polygenic risk score. There's one, let me see if this is mentioned in the other slides that I shared in class. There's um yeah, I think, oh yeah, let me share this one. So, oh, okay.
Okay.
So here, this is showing, I think this one at the bottom is showing some PRS methods that incorporate functional information. Like you mentioned, like this one has functional annotations. Um, but it also can work across populations. So the input to it is if you run a GWAS in multiple populations, it's the summary stats from each of those populations, LD matrices from each of those populations, and then functional information for the variants that you're including. So I think something like this, I haven't used Paraset, and I also haven't used this one, but I've heard a lot of people talking about it, so I think it's promising as well. But um, I think these methods could be pretty interesting and maybe
Okay.
leverage more information than just statistics on its own to try to create a really good polygenic risk score.
Awesome. Thank you so much. Thank you.
Yeah, no problem. Thank you for the question. Oh, and then I see that there is some comment about the RSIDs and base positions and alleles. I'm sure that the R code that someone mentioned will be really helpful and probably the most simple, easy approach to use. A lot of times when I'm trying to figure out the RSID for if I have chromosome in position and I need to know the RSID, a lot of times I'll just annotate using something like variant effect predictor, the VEP software, or Anavar. Those are really good softwares to get more basic information about your variants if you don't have that already. And I think both of them have a web-based tool, so you don't necessarily need to download the software and do it in like a terminal or a Linux interface. You can actually just upload it to the web-based server, and hopefully that would be enough to get RSIDs and other really basic information. Any other questions that came up while you guys were looking at slides, or anything that you would like to discuss that would be helpful today? Okay, well, one suggestion that came up while we were talking about the session was to go over the homework have people already done the homework? And have people also received the answers already, or would it be helpful to go over that right now and go through the answers?
It will be helpful to go over the homework. Thank you.
Okay. Okay, great. Okay, let me see if I can. I'll try to pull up both of these at the same time, to not give away the answers as we're talking about things. But okay, so I'll share the homework first so we can think about it. So I think you guys probably would have done something like this in the first couple of courses or first couple of classes for this course to just know how to calculate a polygenic risk score. But I thought it would be a helpful practice just to reiterate and reinforce what you learned in the previous lessons, just because, I think repetition is often really helpful to make sure that we understand things. But did anyone have any challenges when they were going through this and trying to calculate the polygenic score? This is just a really simple example. There's 4 SNPs here and 5 people, and for each of them, we're trying to calculate their polygenic score given these weights for the 4 SNPs. Did anyone have any challenges, or does anyone want to take a guess at what some of these numbers were? Okay, well, maybe I'll just go ahead and share the answers for that one so that we can see it, and you can maybe just let me know if you got it right. Let me see if there was a chat. No challenges. Okay, good. I'll just share the answers, and hopefully we all have the same answers. Okay. So these were the answers for this. Is this pretty consistent with other people, with what other people got? See if I can. I'm not able to see the chat while I'm in here. Let me see if I can open it.
Some people are saying no. Some are saying yes.
Okay, so some people got these and some people didn't. Okay, I'm glad some people did, because that means I probably didn't calculate it wrong then. So for instance, for this first person, they have 0 copies of the first SNP, so it would be 0 times 0.15, and that's 0. The second one would be 1 times 0.5, and the third would be 0 times 1.25, and then the last one would be 2 times 1. So these ones are the only ones that are actually providing some some quantity to this total number. So it would be 0.5 plus 2 times 1, 2.5. So that's how we arrived to that 2.5 score for this person. And then, for instance, I'll just do one more just to make sure it's really clear. This one would be 0.5 times 1, and then 1 times 1.25, so that would be 1.75. So it's just a weighted sum of the number of risk alleles, and it's weighted by the weight for each SNP, the variant-specific SNP. So SNP1 is weighted by the SNP1 weight here, and so on. So it's a really pretty simple calculation. And you can use tools in like Plink, for example, to calculate these. You don't need to do it by hand. It's nice to be able to do it by hand just so that you really understand like what's going on under the hood of these different software when they're calculating polygenic scores. But it is a really very simple calculation that you can do by hand, and you can program it in R to do it in R without needing something like Plink. But sometimes it's nice to do it both ways just to make sure that you're getting the right answer and you really understand how it's done. Okay, so for the next one, for each of the items, describe a factor that, sorry, are there any questions about that? I'll stop sharing just in case there are comments. Okay. Any questions about that? Hopefully that was something that you guys already went over and you feel good about it already. Okay, so for the next one, I'll look at the answers again. Okay, so for each of the items, describe a factor that impacts the predictability of polygenic score. So here, so select the underlined option that best describes how the predictability can be maximized. So choosing a PGS based on a population with a similar or dissimilar genetic background as the test data. So in the lecture, we talked about there's a few factors that are really important for developing a good polygenic score. And it is important to have a population that's as close to your test population as possible. So when you're developing a polygenic score, you want that training data to have a genetic background that's pretty representative of your testing data as well, so that it will do well in that test population. Second, choosing a PGS developed from discovery data with a small or large sample size. So as we were talking about earlier today, having as big of a sample size as possible in your discovery GWAS is really important for improving the predictability of your polygenic score. And then last, studying a trait with low or high heritability. So here, high is the answer. Traits with low heritability, it's often really hard to develop a polygenic score that is predictive of it because there isn't a strong genetic background for that trait to begin with. So when I was doing a lot of work in cognitive function as a graduate student, there is some heritability to cognitive function, but it's really hard to study it. It's really complex, and it's more than just genetics. There's a lot of things that contribute to it. And there's some other traits that we try to study the genetics of that are just not very fruitful. There's some people that study the genetics of education, and there are a lot of things that play into education besides genetics, and it's very, very muddy and challenging to study the genetics of those traits. Another thing to think about with something like education is what's the goal? Like, why do you want to know the genetics of something like education, especially when you know that there are so many things you can do that are not related to genetics at all to change education and the potential for educational attainment. So just some things to think about as you're thinking about what kind of trait you might want to study the genetics of, is considering the heritability of it, what environmental risk factors are there, and what's really going to make the biggest impact for the trait that you're studying. Any questions for that? I'm going to stop just so that I can see the chat if anyone has any questions. Okay. Thanks. Okay, so I'll go ahead and keep sharing. Let me look at what the next one is first, though. Okay, the next one is describe why polygenic scores often have poor predictive ability in populations that are not included in the discovery GWAS used to develop the polygenic score. So there's a lot of answers to this, but does somebody want to start off with giving some of their thoughts that they had as they were thinking about this question? Yeah, Rachel?
Okay, for me, I thought about the genetic distance between the ancestries as one of the contributing factors. The further the ancestry is from the discovery data or the discovery cohort, further impacts on the accuracy of the PGS, right? And then, and some of the reasons behind this is due to the linkage disequilibrium pattern in between the different ancestries, differences in the causal SNPs, the effect size of the causal SNPs, and also what else can I think of now? I also thought about the differences in environmental factors which may correlate with the PRS. That's a genetic component. So those were the three things that really came to mind.
Yeah, I think that's perfect. I think that really covers like everything. I think, and I also feel like a lot of the times when questions like this come up, LD is often the answer for everything, because that's the big thing that differs between and within populations, and makes it easier or harder to identify genetic variants associated with a trait. So yeah, and I see that Sarah had similar answers in the chat: diet, lifestyle, LD, environment, heritability, and continuity. Those are all great answers. Those are perfect. I don't think I have anything to add to that because that really covers the basis of all the factors. And there could be more. It's also possible that there are more things that we don't know about yet, but those are the big sources right now. And reasons why it's really important to include more diverse populations in our discovery. That way we can ensure that it will apply to more populations when we're testing it. And then the question of LD too. Let me make sure this wasn't another. I think one of the really amazing things about genetics and a benefit of including populations, individuals across diverse populations, is that it can really improve the chance of being able to identify a causal variant. And I think there's an image that I showed of this in the slides. Maybe I'll just pull it up so that we can think a little bit about it, because I think LD is just so important to think about. So here, these are some of the reasons why PGS accuracy can differ. One of them is that the effect of the causal variant may differ between populations, as Rachel mentioned, and then differences in LD structure between populations. So here, you can see that the causal variant has a very different correlation with all the nearby variants in this population as it does in this population. And that could lead to the polygenic score not doing as well in the population that wasn't in the discovery. But if you have both of these populations and you have genetic information, you can leverage information across those populations to try to improve your identification of a causal variant. And the thought behind this is that if a variant is truly causally associated with the trait that you're interested in, you would expect it to be causal in any population. It shouldn't matter what population an individual is from. If they carry a causal variant, you would expect it to be causally associated with disease risk. So here in population A, we see that the strongest signal in this region is this variant. And here you can see what the correlation structure, or the LD structure, looks like for variants that are nearby. But in the same region, you see that this is actually the strongest signal for this population. So, and the LD structure is a little bit different here too. So here in this population, this variant is really strongly associated with the top signal. But in this population, it's really weakly associated with the top signal. So we can kind of safely say that this variant is really unlikely to be the causal variant, because it's not even correlated to the strongest signal in this population. So we kind of expect, just based on these results, that the true causal variant probably lies somewhere between these two signals. And here, this is showing the posterior probability, posterior inclusion probability, the PIP score, just kind of suggesting how likely is it that these might be the causal variant. And you can see that the highest PIP score is really lying between these two strong signals in these two different populations. So you can really leverage information across populations to increase your chances of being able to identify the truly causal variant, which is a really amazing thing about genetics, and an important reason to include diverse populations in our studies. Any questions about that? And then Oscar had a comment. I think different study designs, diagnostic algorithms could cause different results between PRS studies. Yeah, I think that's definitely, so diagnostic algorithms, the way that the outcome is defined is definitely an important thing. The way that the disease or the outcome is defined is definitely important to consider. And if it's different between populations, then the PRS, it might just not be as strong of a marker of whatever trait you're trying to look at, because the outcome is slightly different in one study versus another. And then study designs is an important question too. And there's some trade-offs, because ideally it's great if we could do everything in a prospective setting, but it's harder to get bigger sample sizes for prospective studies if you're interested in a particular outcome. Whereas if you're doing a case control study, you can enroll, for instance, all the prostate cancer cases at a clinic and get lots of prostate cancer cases really quickly, as opposed to enrolling a cohort of men who are prostate cancer free and then seeing who develops it. So a prospective study is definitely a much better study design, but you may have more power challenges to trying to start a study like that, as opposed to doing a case control study design. But it is an important consideration, and it could be an important factor to consider. Okay, so the last question in the homework is, what are the limitations of grouping individuals into distinct population groups in polygenic score investigations? So here there are several different options, and I'll just read some of the options aloud. A is that it ignores, yes, Salma said F. And yeah, that is the correct answer, it's F. So I'll read off all the correct ones. Ignores inter-individual variability within groups. Populations are not truly distinct. Everyone has some degree of admixture. Group boundaries depend on algorithms, which maybe is also could be something that Oscar was getting at too. Group boundaries depend on reference panels, which may not include the population under study. So these are all challenges to grouping individuals into different distinct populations. There's always some amount and some degree of arbitrariness when you're trying to define a group of individuals who are genetically distinct, because everyone has some degree of admixture, and it's really not possible to define a distinct genetic group of individuals because everyone has some degree of admixture. And there is a lot of, reference panels do not include representation from all populations, so they, are never really ideal to be able to do something like this. And the algorithms that we use, they're based on statistics, and statistics always has some measurement of error. And yeah, and then there is also, because everyone has in some amount of admixture, even if you define a group as well as you can, there's going to be a lot of variability within that group that could be important for how well a polygenic risk score does. And that kind of gets ignored when you group individuals. So those were any questions about that topic or thoughts on it? Okay, well, those were all the homework questions that we had from there. I'm happy to talk a little bit more about some of the lecture content if anyone has more questions, or if there's any topics that people were interested in that they'd like to dig into a little bit more. Maybe one thing that I'll highlight, since I think it's probably of interest to this group, but you would have already seen it if you looked at when you watched the lecture. I thought this was a nice review that I came across when I was putting together the lecture material, where the author of this paper looked at the genetic diversity of the polygenic scores that are developed in the PGS catalog. And it was really interesting to see that of the polygenic scores developed, there are a number of them, 87 different polygenic scores, at least include African populations, but there are none that were developed exclusively in African populations, which seems like a big miss. So I think there's a lot more work that can be done to advance polygenic scores and improve their predictive ability. So this is an upset plot. I don't know if people have seen upset plots a lot, but it shows, for instance, like of these 3,200 polygenic scores, they were all developed in European ancestry individuals. There's 125 polygenic scores in the PGS catalog that are developed exclusively in East Asian individuals, and 77 were developed in populations that included both European and East Asian ancestry individuals. So that's just to give you a little bit more idea of how to interpret it. And if there were any that were developed in African populations exclusively, then there would be a dot here and not in any of the other options above it. And you can see that the instances where it does occur, there are dots above it too. So it's always including more than just African populations. And here in this figure, they are distinguishing. African is defined as African sub-Saharan African or African unspecified. So that is a little bit unclear, but there also is a category for African American separate here. But even in this case, you can see there are also no instances of polygenic scores that, at least at the time that this study was done, that exist in the polygenic PGS catalog that are developed in African American populations exclusively. So Sarah has a question: how do you control for LD in PGS calculation? So there's a few different ways that you can do this. When we develop a genome-wide significant polygenic risk score or a polygenic score, for instance, for prostate cancer, we have the 269 or the 451 variants associated with prostate cancer. Those are all independent of each other, or at least independently associated with prostate cancer. So their LD is not playing into at least how the polygenic score is calculated. Of course, there's a lot of challenges with that because LD structure does differ across populations, but for our polygenic score, we developed it across multi-ancestry populations, so those variants are independently associated in each of the populations that we investigated. But for instance, when you're looking at the 1.1 million variants, those LD is more of an issue there for those genome-wide approaches, and that's why they include the LD matrix to be able to account for the LD structure between all of those variants that are included in the polygenic score. And in that case, it would be the LD matrix from the population that you're trying to calculate, or that the discovery GWAS was calculated in. But in that case, for instance, if you're developing a polygenic score that is based on European and African ancestry individuals, so you give it GWAS summary stats from European and African ancestry individuals, and LD matrices for both of those populations as well. Then if you want to go and apply that polygenic score to an East Asian ancestry population, LD structure won't have been accounted for there, so it's possible that there could be some correlation between the variants that is not accounted for properly. But generally, we either limit polygenic scores to variants that are independent of each other, or you can include an LD matrix when you're creating the polygenic score, and when it reweights the variants, it'll be taking into consideration the LD between variants, so you can upweight or downweight to make sure that you're still reflecting variants in an independent way from each other, if that makes sense. Sarah, was that helpful to answer your question? Okay. Any other questions that anyone has, or any topics they'd like to talk about from the lecture? Okay. Well, Dr. Let me see if there's Dr. Rashid, I don't know if you have any suggestions. Should we end a little bit early, or do you have anything topics you'd like to go over?
Very good. I think we're good. Did anyone of you guys have any more questions for Dr. Jars?
And if you think of them later, you're welcome to send me an email. I think I included it in the lecture slides, and we can share that with you too if you have any other questions.
So much, Doctor.
Of course. Thank you so much for having me. It was great to get to talk to you all and have some conversations. And again, please feel free to email me if you have any questions. I'm happy to talk more.
you so much for answering all the questions. It was our pleasure to have you with us. Just one little thing before we wrap up, we would love to capture a group photo if you don't mind. And please, if everyone is comfortable, please turn on your cameras. We will take a small photo for our group. And one last thing, please complete the post-lecture survey. You can find it in the chat box. It will be important to track your attendance. So let's go for the group photo. If you don't mind, just tell me when you take the photo. Yep, I guess we're good. Thank you so much again, Dr. Darst, and thank you all for attending. We hope you found it informative and enjoyable. Have a great day.
Great. Thank you all so much. Bye.
Bye.